Hi, Dr. Vinay. Dr. Vinay Sagar, are you able to hear me?
Still is not a panelist. Arun.
No, I have already made him a panelist, sir. So I thought I will ask him to share and check it once.
Yeah, no, but it is not showing as a host or a co-host. If you look at the panelist list, okay, yeah, he is the right. Dr. Vinod Kumar is also seen as a panelist. I think. Uh,
Oh, yeah.
yeah,
Okay, sir.
yeah.
Dr. Vinay Sagar, are you able to hear us?
Am I audible, sir? No.
Yeah, I think now you're audible. Would you like to just share your slides and check if everything is fine, your audio, video, and the slides?
Sure, sir. Sure, sir.
Hi, Dr. Vinod Kumar, sir. Welcome to the ISN Journal Club.
Yeah. Okay. Thank you. Thanks, Odd. I'm just setting up everything.
Perfect, sir. Take your time, sir. We have still 10 minutes to start anyhow.
Sir, I have shared my presentation
You can
slides.
probably put it in presentation mode and we can wait till 8 and then we can start.
Okay, sir.
Sir, please make a slideshow.
Pardon, sir.
I think we'll make it in presentation mode probably. Slideshow.
Yes, sir. No, it's okay, sir.
Yes.
Okay. Dr. Vinod has still yet to join us. So,
Hello, Darshan.
hello, madam.
Yeah.
Thank you for joining in, ma'am.
Yeah.
We have
So,
Professor also here joining.
Dr. Vivek has also joined.
Yes, sir. Yes, madam. Dr. Vivek, is joined. Doctor.
Yeah, thanks.
Good evening, sir.
Hi, good evening.
Yes.
Hello, sir. Good evening. Good
Hi,
evening,
sir.
ma'am. Welcome.
Good evening.
Can we start the session? It's 8. Okay. Um, Thank you all for joining in. We will continue with our monthly session of the Indian Society of Nephrology Journal Club. This month, we have Professor Jha from the George Institute of India and Dr. Urmila Anand from Amrita Institute, Faridabad, who are joining as the panelists. They will be the clinical panelists who would go through the relevance of the study and whether these things can be useful in our day-to-day clinical practice. We will have critical comments from them describing describing about the presentation from the student and how things can be improved, and also for young researchers, they will also throw some idea about how these studies can be replicated in an Indian cohort. We are also joined by Dr. Vinod today. Dr. Vinod is the basic scientist working in PGI Chandigarh. He has tremendous amount of exposure, especially in bench work, working on podocytes. He has obtained his he has done his postdoc fellow in US, and he's going to talk about the intricacies involved in protein extraction and how the antigens antibodies are identified in the experimental lab. So without much ado, I want to invite Dr. Vivek Sagar, who is DM resident at the RML Institute, Lucknow. Vivek.
Good evening,
Yeah.
respected faculties, seniors and colleagues. This evening, I'm going to present ISN Journal Club for the month of July 2024. Shall I start, sir?
Yeah, Vinay.
Yes.
Your slides are visible, you're audible, you can go ahead. Yeah.
Yes. The title of today's article is Autoantibodies Targeting Nephrin in Podocytopathies. It is an original article published in the New England Journal of Medicine on 25th of May 2024. Podocytopathies are characterized by damage to the podocytes with appearance of proteinuria. Podocytes are specialized epithelial cells of the glomerular filtration barrier, which regulates the filtration process. Histopathologically, the podocytopathies can be classified into membranous nephropathy, diffuse meningeal sclerosis, minimal change disease, focal segmental glomerulosclerosis, collapsing glomerulopathy. We can also classify podocytopathies on the basis of pathogenesis into pervading factor mediated, toxin mediated, genetic mutation related, or hyperfiltration mediated. Factors which are responsible for the development of podocytopathies can be divided into intrinsic factors and extrinsic factors. Intrinsic factors include dysfunction in the transcriptional regulators, or slit diaphragm complex abnormalities like mutation in the nephrin, porosine, or actinin 4 genes, luminal and abdominal protein abnormalities, alpha and beta DG, TRCP6, cytoplasmic protein abnormalities, metabolic causes like Fabry disease, and extracellular proteins alteration, LAMP2. Extrinsic factors, infection is the one of the cause, and particularly the viral infections, toxin mediated and medication mediated. Mechanical causes such as obesity and hyperfiltration. Immunological causes such as lupus nephritis, IgA nephropathy, membranous nephropathy. Metabolic causes such as diabetes. The next question is, why was this study needed? This study was performed to assess the utilization of anti-nephrin antibodies as an early and non-invasive biomarker to establish the diagnosis and monitor the disease activity during the immunosuppressant therapy. To discover appropriate assays for estimating qualitative and quantitative anti-nephrin antibody status, to establish a pathogenic role of anti-nephrin antibody with podocytopathies. Methodology of study: it is a multicentric study conducted in medical centers in Germany, France, and Italy. It is designed as an exploratory study. 239 adult patients were enrolled from glomerulonephritis registry in Germany, and 33 pediatric patients with the idiopathic nephrotic syndrome from Bambino Gesù Children Hospital, Rome, Italy. 118 adult patients and 5 pediatric patients with idiopathic nephrotic syndrome from University of Bari, Moro, Italy. 144 pediatric patients with idiopathic nephrotic syndrome from Paris, France. And samples from the 50 children from the Prince study group were taken as a pediatric control. These are the inclusion criteria in the study. All the patients with the renal biopsy-proven podocytopathies and children were diagnosed with the idiopathic nephrotic syndrome based on the clinical phenotype and lab findings. Recombinant human and mice nephrin was generated from the ectodomain of the human and murine nephrin, and recombinant protein construct were expressed in human embryonic kidney cells. Immunoprecipitation assay was used to determine the anti-nephrin antibody status, and for quantitative anti-nephrin antibody, a hybrid assay involving the immunoprecipitation and ELISA was utilized. And an animal experiment was conducted in which the male wild-type mice were immunized with the recombinant murine nephrin. Serum or plasma samples were taken at the baseline and 2 weeks after the immunization, as well as on the day of sacrifice. Subsequently, mice were sacrificed after an observation period of 3 weeks post-immunization for blood and the organ collection. Because of the explorative nature of the study, analysis did not yield p-values, and clinical data are shown as scattered plots with the medians and 95% confidence interval. To quantify the association between anti-nephrin antibody status, disease activity measured at USCR or UPCR, linear repeated measures model were fitted to natural log transformed urine protein data for adult and children who tested positive for the anti-nephrin antibody and had available follow-up samples and data. Time point with the within patients were treated as repeated measures, while anti-nephrin positivity, sex, as well as their interaction as fixed effects. Correlation strength of anti-nephrin antibody titers with the proteinuria and serum albumin was assessed by Spearman correlation. Statistical analysis was performed using the SPSS and GraphPad prism. Coming to the results, this is the table showing clinical characteristic of adult patients with glomerular diseases. Out of 105 MCD patients, 46 were anti-nephrin positive, accounting for about 44%. In primary FSGS, among 74 patients, 7 were anti-nephrin positive, antibody positive, that is around 9%. And in non-primary FSGS, only 1 was anti-nephrin positive. And the rest of the glomerular diseases did not show any anti-nephrin positivity. This is another table, and this table depicting the clinical characteristic of children with idiopathic nephrotic syndrome. Among the 182 pediatric patients with idiopathic nephrotic syndrome, 94 were anti-nephrin antibody positive, that is 52%. This slide is about prevalence of anti-nephrin antibody in adults and the pediatric patients. In MCD patients, prevalence was 44%. Among primary FSGS, it was 9%. In MCD with nephrotic range proteinuria, prevalence was 60%. And MCD patient with the nephrotic proteinuria before the start of immunosuppressant was 69%. In pediatric idiopathic nephrotic syndrome patient, anti-nephrin antibody positivity was 50%. And among idiopathic nephrotic syndrome patient with nephrotic range proteinuria, it was 80%. And in idiopathic nephrotic syndrome with nephrotic range proteinuria before the start of immunosuppressant, prevalence of anti-nephrin antibody was 90%. This is the scattered plot illustrating the clinical measures in the anti-nephrin positive and anti-nephrin negative MCD and primary FSGS patients. On the extreme left, UACR or UPCR is showing the UACR or UPCR has a higher values in the anti-nephrin positive patient. Second graph is showing serum albumin values are higher in the anti-nephrin negative patient, whereas serum cholesterol are more deranged in the anti-nephrin antibody positive patients. However, estimated GFR in both the groups, anti-nephrin positive and anti-nephrin negative, were in the similar range. This study also evaluated the anti-nephrin antibody status during the longitudinal follow-up of the patients. And 163 follow-up samples were collected from the 38 adult patients. And out of 38, 18% had anti-nephrin positivity during the follow-up at 2 or more point of time. Thus, the presence or absence of anti-nephrin antibody were associated with the active disease and remission respectively. Linear repeated measure models revealed a strong association between the anti-nephrin antibody positivity and the protein levels in the urine. Strong correlation between all measures anti-nephrin antibody levels and the urinary albumin to creatinine ratio with the R value of 0.64. These are the two scattered plots. Plot A is showing basically elevated levels of anti-nephrin antibodies in MCD and primary FSGS patients, whereas it is negative. Anti-nephrin antibodies are negative in the MN and the control patients. Plot B is showing strong positive correlation between the UACR and anti-nephrin antibody levels with the correlation coefficient of 0.64. These two are the heat maps showing a relationship between the antibody status and the disease activity in 18 adults and 18 pediatric patients during the follow-ups. And as you can see in most of the follow-ups, there is a correspondence of anti-nephrin antibody. The corresponding anti-nephrin antibodies status was very much related to the raised albumin or UPCR or UACR values. So in upper bars are actually showing anti-nephrin antibody status during the follow-up periods, and the lower bar of a particular patient is showing the UACR values. That is, red is more than 3.5, pink is 0.3 to 3.5, and blue is less than 0.3. So it is anti-nephrin antibody status has a corresponding to the values of the USCR or UPCR. It is elevated in case of anti-nephrin antibody positivity. Similarly, in the children also, during the follow, it is showing a similar kind of picture. This is a graph depicting longitudinal course of a patient with multiple relapses during the use of steroid and cyclosporine. The black line is showing USCR, and the red line is showing anti-nephrin antibody levels. And after the administration of rituximab, there is dramatic fall in the level of both USCR as well as the anti-nephrin antibody level, and patient then went into remission. A similar kind of graph with in a patient, in two patients, patient A and patient B, who were using prednisolone and cyclosporine, and after they received the rituximab, there is a dramatic fall in the both the USCR as well as the anti-nephrin antibody level. On this study, they have replicated a mouse model, and they replicate a podocytopathic phenotype in the experimental mouse model, and they have actively immunized mice with the ectodomain of the murine nephrin, and thereby induced the anti-nephrin antibody production. And this resulted in the rapid development of nephrotic syndrome with the high UACR, reduced serum albumin, and increased cholesterol level at 3 weeks post-immunization. These are the histological picture of the control and nephrin immunized mice. Uppermost figures are light microscopy staining with the past periodic acid skiff, and in the control as well as in the nephrin immunized mice, there is no, both are unremarkable. The middle pictures are electron microscope picture in the control and nephrin immunized mice, and in the control there is the food processes are intact, whereas in the nephrin immunized mice there are diffuse food process effacement. The bottom one is immunofluorescence, and they have labeled the mouse IgG with immunogold, and it revealed the localization at the slit diaphragm in between the effaced food processes. And there is also nephrin redistribution from the membrane to the cytosol, and a fine punctate pattern of mouse IgG positivity, partially co-localizing with the nephrin. Proteomic and phosphoproteomic analysis were also performed to test direct effect of anti-nephrin antibody on nephrin signaling and downstream pathways. They have quantified 10,545 phosphosites, 5,696 of which differed significantly between the control and immunized mice, and phosphorylation of nephrin was increased at the tyrosine residue Y1191 with the log2 ratio between nephrin immunized and the control of 1.053 and the p-value of adjusted p-value of 0.0. This is the diagram illustrating the phosphoproteomic analysis of the phosphorylated tyrosine residue Y1191, and it is clearly showing greater phosphorylation of tyrosine residue in the nephrin immunized mice as compared to the control, with the p-value of 0.02 and log2 ratio of 1.053. Thus, after increased nephrin phosphorylation, there is increased SSCA and clathrin at the podocyte membrane, strong down regulation of the core slit diaphragm, associated proteins, and changes in the podocyte adhesion proteins, and actin binding or cytoskeletal proteins. Thereby, these changes leads to the loss of functional slit diaphragm and and there was production of the MCT-like phenotype. Coming to the discussion, this study has investigated the prevalence and the clinical significance of anti-nephrin autoantibodies in adults and childhood onset glomerulopathies. And anti-nephrin autoantibodies were present in 44% of adult patients with MCD, 9% of those with primary FSGS, 52% of children with idiopathic nephrotic syndrome. Thus, defines this condition as anti-nephrin associated podocytopathies. The prevalence of antibody was 69% and 90% in MCD and idiopathic nephrotic syndrome with active disease before immunosuppressive treatment given respectively. These are the previous studies conducted to know the prevalence of the anti-nephrin antibody in the podocytopathies. The prevalence ranges from 29% to the 46%, although the sample sizes were small in these studies. One of the studies showed a significant high level of anti-nephrin antibody in the MCD and primary FSGS patient as compared to the other glomerular diseases. Study by the Shirai et al. Demonstrated high anti-nephrin antibody level in the post-transplant recurrence of the primary FSGS. Anti-nephrin autoantibodies as a new reliable biomarker for monitoring disease in the anti-nephrin associated podocytopathies. 3 of the patients with the anti-nephrin associated podocytopathies on rituximab turned anti-nephrin antibody negative and attained clinical remission. This suggests rituximab-induced remission in patients with the nephrotic syndrome may be a consequence of autoantibody depletion. Immunoprecipitation assay, which enriched IgG, facilitated the specific and reliable qualitative identification of anti-nephrin antibodies. Newly developed hybrid technique, which combines immunoprecipitation with the nephrin ELISA, allowed for the quantitative measurement of the anti-nephrin and autoantibody levels. Thus, provide a non-invasive alternative for the diagnosis, enhancing treatment evaluation, and improving kidney transplant strategies in patients with the recurrent primary FSGS. Pathogenicity of the anti-nephrin autoantibodies was illustrated by the development of anti-nephrin autoantibodies and nephrotic syndrome in mice that were immunized with the nephrin. Anti-nephrin autoantibodies induced phosphorylation of nephrin at the tyrosine Y1191, a process critical for mediating nephrin endocytosis and maintaining podocyte cytoskeletal integrity, which resulted in profound actin and cytoskeletal alteration without triggering transitional immune responses. So the conclusion of the study is that the antibody-mediated podocytopathy is a newly diagnosed, newly discovered entity. Anti-nephrin antibody targets nephrin, disrupt kidney function without triggering traditional immune responses. Anti-nephrin antibodies can be considered as indicator for disease activity and has a direct pathogenic role in antibody-mediated podocytopathies. These discoveries open a new avenue for further prospective studies and pathologically tailored medical interventions. Limitations of the study are exploratory nature of the study. Since exploratory studies provide qualitative data and interpretation of which can be judgmental or biased. Second limitation of the study was a small sample size. And third one was there was no information about the race or ethnicity in the adult subjects. And the lack of standardized and validated assays to detect the anti-nephrin antibodies. Strength of the study was longitudinal in nature, and they have measured the anti-nephrin antibody and several follow-ups and different point of times, and the use of model to establish the pathogenicity of the antibody, the use of proteomics and phosphoproteomics to assess the downstream effect of pathogenic antibody, and they developed a immunoprecipitation ELISA hybrid assay for the quantifying anti-nephrin antibody level, although it was not validated in any other studies. Thank you.
Thank you, Dr. Vinay. Dr. Vinod,
Yes.
yeah, you have your slides, or Dr. Vinod, do you have your slides with you, or you would be just taking through the experimental study?
Just with the experiments.
Okay, so I'm just sharing. Good evening
Yeah.
to everyone.
Oh, I think we have with us Dr. Vinod Kumar. He'll be explaining to us about, in general, the experimental models which were used in this study, and in general, what goes behind in the lab. So after that, we'll have a discussion wherein we'll invite Jasar and Urmila ma'am to take it forward, discuss the presentation per se, and about the implications of the study for future research. I would request the attendees to post their questions in the chat box, and we can take it up for discussion also. Dr. Vinay, I think you can stop sharing your slides, and Dr. Vinod Kumar sir will then present the basic parts.
Yes.
Just to introduce Dr. Vinod. So Dr. Vinod has done his PhD in stem cell and mitochondria from the PGI Chandigarh and completed in 2014. He was working as a project scientist at the George Institute of Global Health, and then he did his postdoctoral research fellow from New York, and he continued to work as a senior research associate in New York until he joined as an assistant professor back in PGI Chandigarh from 2019. So Dr. Vinod will take us through the bench work and the experimental model that was used in the journal. Dr. Vinod.
Thank you. Thanks a lot. My screen is visual.
Yes.
Yeah. Okay. So today I'm just discussing that how they have designed this particular assay that they call the hybrid ELISA. So on this screen, I'm showing one side is the recombinant nephrin, that is the two green color tags. One is the twist strap tag and one is the his tag. And the other side is the right hand side is the anti-nephrin antibody. So they have used this recombinant protein with these two tags to isolate the circulating autoantibodies from the serum. So this whole story or whatever they have designed is a move around these two tags. So these are the method they have used in this study. So I am here just talk about the three methods. First is the immunoprecipitation, second is the quantitative immunoprecipitation ELISA assay, and the third is the anti-nephrin immunoprecipitation Western blot. Yes, sir.
Can I ask a question?
Yes, sir. Yes, sir.
If you go back to the previous slide.
Yes.
Because this is predominantly audience of clinicians. Can you explain what is this his tag and the twist trap tag?
Yeah, sir, his tag is a histidine. They have a consecutive amino acid of histidine, like 6 to 7.
Seven
And
amino acids, right?
Yeah, yes, sir.
Yeah.
Yes, sir. 6 to 7 or 8, it can be extended. And the twist strap is a streptavidine. So, there is a two bands of streptavidine that are tagged with the protein.
And why do we use these tags?
Sir, the streptavidine they have used for this protein, this is you can say the USP of their design.
Right.
That I
So
can explain in the next slides.
These tags are used to identify the protein. If you,
Yes,
because
sir.
tags are easy to identify. So
Yes,
it is
sir.
like someone wearing a cap, right? So if you have one team which is wearing red cap, if you only identify the red color cap, you know that this person belongs to this team. So similarly, if you identify a tag, you know that this protein which is linked with that tag must be there, right?
Yes, sir. Yes, sir. Because this antibody binds to the nephrin. This recombinant nephrin part. But we can use these two tags to identify or amplify the signal of this complex, the antigen antibody complex, by
Correct.
using these two tags.
So if
Yes,
you
sir.
Does it increase the avidity of binding in any way?
No, Urmila, don't.
Or it's just a specific antibody
Urmila, just
binding?
don't get confused. These are not antibodies. These are tags. This is like a flag. When you go around,
Histochemistry, the kind of a stain.
when you go to, listen to me, when you go to a tourist spot, you are in a group and you have a guide and the guide
Flag
carries
kind of
flag.
thing. Yeah.
So that is it, so that you know that this, when you see this flag, you know that this antigen antibody complex is
Has
present.
happened.
Nephrin antinephrin complex is present.
Yes, sir. Yes, sir. The recombinant protein can also be expressed without these tags, so it depends, like, what is the demand of assay, what type of assay you want to design or you want to perform. So they have used these two tags, and they have used these two tags to, validate all their studies. So, like, if I'm just talking about these three assays because they failed to detect anti-nephrin antibody by using the conventional Western blot. The Western blot is where we are, that electrophoresing nephrin protein and then incubating that membrane with the serum of the patient. They did it, like in the PLAR or some other uh, antigen or antibody identification. They use the Western blot, conventional Western blot, to identify the autoantibodies. But in this case, they tried it, but they failed to find any positive results. So that's why they adopted for the immunoprecipitation assays. So, this is what they have done. They firstly use the recombinant nephrin that don't have any tags, not streptavidin tag, not histidine tag. And they run Western blot and they couldn't find any band. They are unable to detect any antibody. They have used the control individuals, and these are the uh, uh, MCD patients. The R showing the uh, denaturing condition, and this is the normal non-denaturing conditions. So they are unable to find any positive signal over here to check whether they have a good amount of antigen loaded in the Western blot or their gels. They have done the simply they have incubated with the anti-nephrin antibodies that are commercially available, and they are able to see the bands. So this picture shows that uh, there is the expression of recombinant protein, but these proteins are unable to detect the autoantibody present in the serum. So what they did, they adopted for immunoprecipitation assays. So what they did, they have developed, they have named it as a quantitative hybrid IP immunoprecipitation ELISA or immunoprecipitation Western blot assays. So what they did, they combined the two techniques. One is the immunoprecipitation and the other is either ELISA or either Western blot. So immunoprecipitation is simply like the precipitation of the immune complex, like the antigen antibody complex, from a circulating media to some solid form. Like we have some antigen and we have some antibody in the serum. We will mix these two, they will form a complex. Then we will use certain beads that we call agarose bead, that they have IgG binding sites, and they can by using centrifugation they can be pulled down because they have some weight, and they will make a pellet after the centrifugation, and then we do the elution and we can find the antigen and the antibody both, and we can use these two for either for ELISA or either for Western blot. So they have used this technique, and then they deployed it for these two assays. So for the immunoprecipitation, what they did, they firstly overexpress the recombinant nephrin protein by using only the his tag. They didn't use here the twist trap tag. They just used the his tag, the first step they did, and they incubated these recombinant protein with the serum of the patient. And similarly, like in spite of using agarose with IgG, for the his tag we can use nickel resin. They can also bind and they can pull it out, and they did the immunoprecipitation, and they have run this on the Western blot. So here, the first, second, three, the MCD patient, FIGS, MN, IgN, and control. They have run the Western blot and they are able to find the antibodies. So, what they did, that by doing this experiment, they have enriched the antibody. Means, in spite of running the antigen on the Western blot, they firstly incubate the antigen with the antibody, so that the maximum amount of antibody can be pulled out from the serum. Just to concentrate more and more antibody, they have adopted this technique, and they were able to find the positive Western blot. Then they use the same recombinant antigen, the nephrin, where there is a no expression of twist trap tag, and they did the ELISA that we call the conventional ELISA, in which they have coated this nephrin protein and then incubated the serum of the patient and then tried to detect. Were they unable to find any positive signal over here? So what they did, they introduced a twist strap tag. So they have used this twist strap tag here, and then they have, like for the ELISA, like we can say these type of wells. So they have used these wells. They have coated these wells with streptactin that bind with the twist streptin tag, and they enhance their binding of this protein. So after doing this, this is the without twist strap tag, and this is the with twist strap tag, and they are able to differentiate the the antibody, the positive expression of the or the positive presence of the antibody in the serum. So the use of these two tags, the his tag they have used for the Western blot, and the use of this strap tag uh, help them in detecting the auto antibodies in the ELISA, which they are failed to detect by using the Western blot. So this is the two experiments they are mainly used to detect the autoantibodies to the nephrin. And here I'm just trying to tell that how they have designed this recombinant nephrin protein. So they have used this type, the ectodomain of the nephrin, which contain 8 ectodomains, and fibronectin domain. They isolated the total RNA. Actually, this whatever the nephrin clone, the recombinant protein they have used, they got it as a gift from someone. But how they have isolated, they isolated the total RNA from the kidney tissue and then converted into the complementary DNA. And by using the simple PCR, they have amplified the nephrin gene. And they have then, by using the PCR, we can add these tags to the proteins. And they have done this. They have added this tag to the protein, and they also added a leader sequence. This sequence help in the secretion of this protein. But what happens that whenever we overexpress any protein, it mostly get retained inside the cytosol, and we have to lyse the cells and then take out the protein. But if we can add this sequence over there, then the protein automatically come out into the media. So then they have cloned this nephrin protein into the expression vector and transfected into the human embryonic kidney cells. And from here, this protein gets secreted into the media, and they have isolated this or they have precipitated this protein from the media and used for like Western blot and the ELISA. They have used polyhistidine for the protein purification, and mostly for the Western blot, and the twist step tag for the ELISA they have done. So this is the quantitative hybrid IP ELISA. So it's the same thing they have done, but this is the here we can see the readings they have reported over here. They try to make it a quantitative ELISA because they quantified the amount of protein. So for that they have designed they have performed one standard curve analysis by using the protein and then the different concentration of nephrin, and they uh, here like in this table there is a two reading. One is the immunoprecipitation reading by using the Western blot, and other is the immunoprecipitation ELISA. So they have confirmed all these by using two methods, that they are positive in the immunoprecipitation and they are also positive in the ELISA, and they have titrated all the autoantibodies. Thank you. I think we are finished with this.
Thank you, Dr. Vinod, for the presentation and going into the intricacies of the molecular methods involved during the process of this study. So the Journal Club article is now open to comments. Please write your comments in the chat box, and the panelists will take up your comments. So to start with the discussion, I would, now request the panelists to give their opinion on, that given the fact that MCD, FSGS, and the idiopathic nephrotic syndrome that we have in childhood are common diseases in nephrology clinical practice, and this study, together with the the background that we had over last few years, which suggested the association of anti-nephrin antibodies with this clinical phenotype of nephrotic syndrome. Looks like that we now have a promise of a biomarker which could associate with, diagnosis as well as response to therapy in such patients. So what is the take of the panelists on this? Like, where do we go from here now?
Professor Jha, would you like to go ahead?
You go ahead, Urmila, I'll follow you.
I just have as the Journal Club article was presented, the first thing is there are a lot of manipulations done to the nephrin antibodies to sort of, catch and nephrin antigen to catch hold of the antibodies. How does this, I mean, I did not, there have been many studies before. How does this study compare with antibodies of detected by various other studies which the Dr. Vinay had put in, where they have looked into nephrin antibody levels, and they have tried to identify. That's one. The second here for me was the diagnosis of the use of this antibody for diagnosis doesn't seem to be that important as much as in follow-up. They have shown that the levels like ANCA, they wax and wane along with the disease. And there was no mention to say that the antibodies actually come down before the disease resolves, except in one part of the paragraph. And the third thing is our concern in INS always has been, in children especially, is this INS going to turn out to be steroid resistant? So I don't think this antibody has helped us with any kind of a prognostic importance as far as diagnosing INS in children. Adults, they have done MCDs, FSGS, and they have shown that the antibody is more seen in minimal change, and in non-minimal change disease, hardly any anti-nephrin antibodies have been detected. So that's how I look into it, and I will invite comments from Sir and from other moderators also as to what is their take on this study.
So thanks, first of all, thanks, Dr. Vinay Sagar, and thanks, Vinod, for presenting the essential findings of the study, and to Vinod for sort of unraveling the methodological aspects of how these assays are done and what do they mean. I would take you a little bit in the history of this. We all know that minimal change disease responds very quickly to steroids. And as a result of that, we have often said that there must be an immune-mediated mechanism. But until recently, when we did kidney, the name minimal change disease itself suggests that we didn't find anything in the histopathological examination of the tissues. And it was only in by ultrastructural examination that we found food process effacement. Immunofluorescence was typically described as pristine. But I think the point to be made here is that we should always be on the lookout for whatever is out of the ordinary here. And if you read some older descriptions, Dr. Helmut Renke, who is the nephropathologist at Brigham's and Women's Hospital, Harvard Medical School, he and his lab, they have described some punctate IgG staining from time to time in children with minimal change disease, suggesting that possibly, immunoglobulins are involved in some way or the other. Then subsequently, fast forward, a number of clinical observations were made. For example, in 1998, we realized that nephrin is a protein that becomes abnormal in minimal change disease. And that in the congenital nephrotic syndrome, nephrin deficiency leads to congenital nephrotic syndrome. And these children with congenital nephrotic syndrome develop kidney failure. They do get transplanted, and many of these children with congenital nephrotic syndrome, after transplant, they developed a recurrence of the disease, right? Quite clearly, it could not have been recurrence of the basic disease. It was the recurrence of nephrotic syndrome. And when the investigators, and naturally this is somewhat akin to what we see when we transplant patients with Alport syndrome, that they develop anti-GBM disease, right? They can develop anti-GBM disease, or they develop anti-GBM. They don't develop anti-GBM disease, but they develop anti-GBM antibodies and staining. And in this case, then they subsequently discovered that these children with congenital nephrotic syndrome, and due to nephrin deficiency after transplantation, they were developing anti-nephrin antibodies. And of course, research was going on in the lab of Helmut Reinke and others. And in that research, they showed that when mice, they were injected with antibodies, just like has been shown in this current paper, they developed nephrotic syndrome. So by that time, it was clear that antibodies to nephrin can give rise to development of nephrotic syndrome. Subsequently, as Dr. Vinay Sagar has shown in a few publications also, that this association was established in humans with nephrotic syndrome. So I think the fact that anti-nephrin antibodies can cause minimal change disease in nephrotic syndrome is not a new discovery, but it has been known for some time. The challenge was 2 or 3 folds. One was, as Vinod has said, and as Dr. Vinay Sagar has also said, that identifying this antibody was really very tricky. There was no good assay to identify these antibodies. So in this paper, I would say one of the most important findings of this paper actually is the description of the assay, that they have developed a reliable assay to be able to develop, to be able to detect these antibodies in circulation. Because going back to the point of whether this will be clinically useful, I think to be clinically useful, we need to have an assay which is reliable, which can allow us to detect the antibodies with a fair degree of certainty, just like we have with now the anti-PLA2R assay, which allows us to follow the antibody titers and then to make a judgment of the course of the disease so far as immunological activity is concerned. That is number one. We are still some way from there. We don't have an assay which can be utilized for clinical use. For example, there is no commercially available assay yet. It is still in the lab. But then, we have made an important breakthrough and we will get there hopefully. That is number one. Number two is our ability to understand the pathogenesis now of minimal change disease. If you looked at the graph that Dr. Vinay Sagar presented very carefully, more than 90% of children with idiopathic nephrotic syndrome who had not yet received steroids showed these antibodies in circulation, at the time of diagnosis. So it does seem to suggest that an overwhelming majority of pediatric nephrotic syndrome is definitely associated with this and may even be caused by this. And that is something which remains to be seen in clinical trials. What implication it has? The implication it has is just like for membranous nephropathy, it is now possible that therapies that target development of these antibodies, such as anti-B cell therapies like rituximab or other anti-B cell targeted therapies, might become the preferred modalities of treatment in this condition. I'm only saying mild because all of this is still in the realms of speculation and needs to be tested in clinical trials. A number of other requirements do need to be fulfilled. Number three is an improvement in our understanding of nephrotic syndrome. Now, it's easy for us to say that, yes, this just because the antibody is present, it causes nephrotic syndrome. Fine. But if we all know the clinical behavior of minimal change disease, you start a child with minimal change disease on steroid, and the child becomes non-nephrotic in 1 week, right? Or even earlier, most of the children. 1 week, 2 weeks, something like that. They don't go more than that. That is not something which is consistent with how an antibody-mediated disease should behave, because the half-life of immunoglobulin G is 3 weeks. So if that immunoglobulin deposit on the nephrin, slit diaphragm is leading to development of proteinuria, then how does it go away in 1 week or 10 days? So that is something which we need to understand better. So there it is possible that there may be 2 or 3 things happening. One is that it is possible that there is some sort of genetic background. For example, you have the ApoL1 gene. We understand all of that. It is possible that there is there might be some genetic abnormality in the nephrin gene, etc., which still remains to be discovered. That's number one. The second is that there is a development and deposition of these antibodies, right, which then lead to a clinical phenotype. And the third is how does the antibody disappear? So it is possible that rather than for the antibodies disappearing just because the antibody production stops, that the antibody might be getting internalized into the podocytes and might be getting shed into the urine, right? And that is something which we'll need to study more by doing appropriate assays, which causes the antibodies to go away much faster than what the normal half-life would lead us to predict, right? So that the antibody goes away by another means. So someone is sweeping away these antibodies rather than these antibodies simply dying because their lifespan has become over. One question which we have not appropriately touched upon, but is there in this paper, is the issue of FSGS. For a long time, it has been thought that FSGS and minimal change disease are part of the same spectrum. That on one hand you have minimal change, on the other hand you have FSGS. That is not so straightforward now. It is possible that the determination, now it is clear that at least a subset of patients with FSGS do have anti-nephrin antibodies. We have also, there has been earlier publications. There is a JASN publication earlier, which has, Kidney International publication, which has shown that anti-nephrin antibodies are present in recurrent FSGS. And that is where plasmapheresis might help, So it is possible that the development of focal segmental glomerular sclerosis also depends on the reaction of the podocyte to the presence of these antibodies. How do the podocytes react to the presence of these antibodies? And that reaction of the podocytes might also be affected by other parameters, biological parameters, genetic background, and so on and so forth, right? So, for example, here ApoL1 might be important because a lot of African Americans who have ApoL1 gene abnormalities develop FSGS. So what I'm trying to say here is that there are a lot of other hidden gems in this paper. So it's not simply straightforward, a paper that looks at patients with minimal change disease having these antibodies and showing some sort of correlation between the presence of antibodies or not. Now, how will it be useful in clinical practice is still somewhere in the future, but I think this is a very, very important advance in our ability to at least develop an assay. And hopefully this assay will become commercially available. I'm sure that there are big pharma already engaged in development of these assays. So this is really very, very important. So those were some initial comments I would make. I see that there are a number of questions in the chat, and people are already jumping to asking any specific titers for positivity of anti-nephrin levels. So Harshil, I think you need to hold your horses. We are perhaps several years away from reaching there, where we'll be able to measure the antibody and say that this titer is clinically relevant, this titer is not clinically relevant. There's one question about confocal microscopy in clinical practice. How is it different from routine immunofluorescence technique? And maybe I can invite you, Vinod, to talk about how confocal microscopy works. So confocal microscopy is certainly very different from immunofluorescence. I would encourage you to actually go and read up about it, Kishan, rather than simply to ask this question and try to take an easy way to understand the answer. You should be able to find the answer very easily. And it's not something which is, it's a completely different way of imaging, right? So you can't compare it with immunofluorescence. Electron microscopy has a different methodology, different goal, etc. But Vinod, do you want to say something about confocal microscopy? How does that work?
Yeah, sure, sir. Thank you, sir. So, confocal microscopy is specifically we are focusing on uh, on a one particular fluorochrome, and here we are using lasers, and what are the fluorochrome we are using? Like if there is a very less expression of one protein, by using the laser we can enhance that very high compared to the fluorescent microscope where we are use the lamp or LED lamp. So they don't have that much of capacity. And other thing is the resolution. So confocal microscopy is the highest resolution, so that we can, like when we are talking about the co-localization or the like the punctate type of features, so we can visualize those stainings very accurately over there. And if two proteins are lying together, we can detect them also. And there is a very huge application of confocal microscopy that is beyond the simple fluorescent microscope.
Yeah, I think the essential feature of confocal microscopy is that it allows you to look at a very, very narrow plane of focus. So you are able to detect proteins or molecules which are otherwise not possible to detect by other microscopic techniques. So as Vinod said, it uses light from a laser. It's a kind of standard light microscope. It's very, very fancy light microscope with a number of other mirrors and lasers and combinations built in. But essentially what it allows you to do is to focus on a very, very narrow area and detect protein which are present in very small amounts also.
Sir, one thing I can add more, like if you want to do some quantification, so we can do it by using confocal microscope, but it's not possible by using simple fluorescent microscope also.
Yeah, thank you, Vinod. So, Dr. Rangaswamy, I didn't understand your question. Can you please clarify what exactly did you intend when you put in that question? What properties?
I think you partially answered it while you were explaining about what the future holds for. Um, while in minimal change disease or FSGS, we don't find an antigen antibody complex. You said some of that antibodies could be internalized and could be probably come out through the urine. Probably that could be an explanation why we don't see antibodies on the split dap on the IF in case.
Yeah, that could be one mechanism, but there could be other mechanisms also that they may be hidden. So, some not every molecule. And again, here, this is where perhaps confocal microscopy can help, as Vinod is saying, because we might be able, because it's, the quantities may be very small, who knows? We don't know what is the degree of affinity of anti-nephrin antibody to nephrin protein. I'm sure it is known in vitro. For example, there are studies in which people have shown that anti-nephrin antibodies do bind to cultured podocytes. So it is not that they don't bind, but in vitro versus in vivo, the binding may be different. Sometimes the binding may be hidden. So therefore, when you actually do a confocal microscopy, you can go very narrow plane by plane by plane, and it is possible that we may be able to detect that. I'm pretty sure that people would have done it, and if it was possible to detect it, somebody would have published it by now, or would be perhaps in the process of publishing it.
Sir, if I can add one more thing over here, like like if we see the literature, so nephrin is well-studied protein in the podocytes. So the most preferable mechanism is the endocytosis. Like other mechanism are also true, but is right now if you go through the literature, so the strongest it might be like when it binds, it get phosphorylated and it get endocytosed along with the antibody.
Yeah. That is why I mentioned it right in the beginning, that it is possible that it gets internalized and then it gets degraded. We also know now that these anti-nephrin antibodies actually lead to redistribution of nephrin, which is identical to that which is seen in normal biopsies as well. And it's something which is similar in culture porocytes and animal models too. So all of that. So it's all coming together, this is the first brick in a wall, and the wall will be built. I think we need bricks, we need cement, everything. And the cement will be the assay, once, if we have a reliable assay, then we'll be off on our way, right? And then it is possible that we will have a true biomarker, just like we have anti-phospholipase 2 receptor antibody. Urmila, sorry, I've been sort of dominating the conversation.
That's
Apologies,
okay.
it wasn't.
That's okay, sir. No issues. But sir, the MCD is just not a B cell disease. There are a lot of T cell activations, and there is a lot of literature to say an immune system cannot work in, exclusivity. Like and that the reason that you said key, even though the antibodies are for 3 weeks, but the response happens within 5-6 days. So there is much more to, I think, B T cell interaction, which we have to understand in the future. So even if you look into plateau R, I mean, steroids don't work. I mean, there are many other things in membranous also. We need a lot more than drugs. So I think the presence of nephrin will help us with some pathogenesis. And as you said, it may divide a nephrin positive MCD and a nephrin negative MCD kind of a thing in the future. I mean, it may sort of categorize MCD much more better. And also, there is a literature in Kidney International just last month where FSGS, a primary FSGS, has been treated with Plex. And they have been measuring anti-nephrin antibodies, and they have shown that this was a case report, of course. As the antibodies came down, the FSGS actually improved. An anuric patient who was Plex. So this paper actually says that they're trying to separate MCD with nephrin and FSGS not with nephrin. I mean, nephrin has not been seen in any of the primary FSGS except one patient in their study.
Well,
So your comments.
I think this is something which is again evolving, right?
Yes, sir.
FSGS, we know that FSGS by itself is not a disease. There's a pattern of injury. It's a light microscopic pattern of injury. And it is likely a conglomeration of multiple heterogeneous conditions. One of which could be anti-nephrin antibody mediated damage. And even there, anti-nephrin antibody mediated damage, what exactly leads to development of glomerular sclerosis rather than only minimal change? And that is what I was alluding to a little bit earlier when I said that a lot depends on how the podocytes respond to
Behave with that, to the
behavior.
antibody or the
Possible
injury
that,
kind of a thing.
and that there could be some genetic background, there could be something else, some other second hit, third hit. We now, in most glomerular diseases, we talk of multiple hits. So all of that might be happening, but that is something which is going to come to us in the future. All the answers are not there today, but we have made certainly a fair degree of advance, paper by paper. You gave the description of the FSGS and post-transplant FSGS, also anti-nephrin antibody, all of that. So I think the bricks are, like I said, coming together.
Or maybe so we will have more antibodies which will add up to the basket like membranous. Who knows? As we understand podocyte biology.
Once the dam breaks and once Sanjeev Sethi brings in his
mass specs,
laser capture microdissection and mass spec, we will identify more. So who knows? It's an opportunity for you, for people on this call to actually start doing it. If you have access to laser capture, it's pretty common. Laser capture microdissection is available everywhere. Mass spec is pretty standard. And so if you are enterprising enough, you have lots and lots of minimal change biopsies available also. So maybe someone in this group will actually take this on. think we are already past 9, Arun. I don't know how long they want to go on.
I see it was till 9 o'clock, sir, and thank you. I think you had taken up all the questions yourself, sir. It was really nice. All the questions were clarified, and I guess I'd like to thank Dr. Vinay for the presentation, Professor Jaz sir, Dr. Urmila ma'am, and Anand ma'am for taking the General Club in a very nice manner, and Dr. Vinod Kumar for such a wonderful presentation on basic sciences. And I guess we are going to have more publications on nephrin antibodies, which will improve our understanding and probably answer all the questions which were in the chat box today. So on behalf of the General Club team, I'd like to thank Jas sir, Urmilanand ma'am, and Dr. Vinod Kumar sir, and Dr. Vinay Sagar, and all the attendees for taking part. And we'll be meeting again on the third Wednesday next month with another interesting discussion. Thank you all.
Thank you very much.
Thank you. Thank you. Thank you, sir. Thank you.
Thank you, sir.